Thanks for joining us today for this UPCARA webinar. My name is Ellen Cichevsky, and I'm from Samba Scientific, and I'll be the moderator today. We're joined by 4 speakers who will discuss an alternative method to lyophilization and frozen cold storage for biomolecule stabilization. Our speakers today include Ron Bowsher, Chief Scientific Officer and Partner at B2S Life Sciences, Alyssa Cieslak, Scientist at B2S Life Sciences, Gabriela Pikul, Scientist at Assay Technologies, and Mary Retzlaff, Chief Scientific Officer at UPCARA. We look forward to reviewing capillary-mediated vitrification stabilization technology for a couple of example applications, including antibodies and kinase assays for compound testing. Without further delay, I'll turn the time over to Dr. Mary Retzlaff to get us started.
Thanks, Ellen. So as Ellen said, my name is Mary Retzlaff, and I'm the Chief Scientific Officer at UPCARA. Prior to joining UPCARA, I worked for about 20 years developing analytical methods for vaccines. And in particular, we were developing potency assays. One of the challenges we ran into very regularly was trying to figure out how to store critical reagents and samples in order to make sure that they were stable and that we could get high quality results for our assays. And if you work in life sciences, I'm sure you've had similar challenges, and you're aware that most biomolecules are temperature sensitive. And that really has an impact on where we can do science, when we can do science, the cost of the work we want to do. And if we don't properly store our biological materials, both the samples and the reagents, it can really impact the quality of the data that we generate and ultimately the decisions we want to make based on our science. So at UPCARA, we recognize that there's a need for newer stabilization technologies, alternatives to both freezing and lyophilization. And as a result, we're developing a new technology that we call capillary-mediated vitrification as an alternative way to stabilize biomolecules and allow researchers to store them under ambient conditions. Our process uses a combination of a proprietary stabilization buffer, a material that we call a scaffold, and vacuum drying to transition biomolecules that are stored in a liquid formulation into a stable glass, or what we like to call a bioglass. The term vitrification simply means transitioning something from a liquid to a glass. And currently today, the best 2 ways to do that are freezing or lyo, but we're going to show you a third opportunity. So our process, as I said before, has 3 components that work together synergistically: the buffer, our scaffold, and the vacuum. And each of these components has a particular role in the process. So the molecule of interest for the first step of our process is blended with our stabilization buffer. The stabilization buffer includes a mixture of inert excipients that very rarely interfere with analytical methodologies. The stabilization buffer has a couple of roles, but the most important function of the stabilization buffer is really to replace the hydrogen bonds that are lost when you remove water from a biological system. Most proteins and most complexes rely on hydrogen bonds to maintain their 3-dimensional shape. When you dry the material, you can lose that 3-dimensional structure, and that can have an adverse impact on the function of that biological molecule. So it's really important to include appropriate stabilizers and excipients that will help maintain the structure of that biomolecule and thereby its function. Once we've formulated the material, we apply it to a porous material that we call a scaffold. You can think of the scaffold as being very similar to a paper towel or a napkin. Those aren't the materials that we're using, but it gives you a good visual. When you apply the liquid to the scaffold, the liquid will penetrate into the pores, and there'll be an interaction between the liquid and the surface of that scaffold material that creates a very high surface tension. That surface tension is really important to our process. If you take a typical liquid and you put it in a vacuum, that liquid's going to boil, and the boiling action will damage sensitive biomolecules. By applying the liquid to our scaffold and creating this high surface tension, we can now put that liquid into a vacuum and dry it without first undergoing a freezing step. And then the final step is the vacuum drying. We leverage vacuum drying to very rapidly remove the moisture and transition from the liquid state to the glassy state. By drying rapidly, we can prevent both phase separation and crystallization, both of which can damage sensitive biomolecules and frequently occur both during freezing and lyophilization. And at the end of the process, we're left with our stable bioglass, which can typically be stored at room temperature. Just to show you how the process works and the simplicity, on the top, I'm showing you how we prepare our stabilized reagents. We simply mix the reagent with the formulation buffer, dispense it, dry and vacuum, and then we package the scaffolds in moisture-resistant packaging. On the day of use, the scientist simply opens the package and rehydrates the scaffold with water or buffer. We can integrate those scaffolds into a variety of form factors, which I'll show you in a minute. And once rehydrated, the material is ready to use in less than a minute and can be applied directly to your assay. Because the process is very flexible and can accommodate a wide range of biological masses, we can select the concentration of each reagent to match what's needed for the assay. So we can store reagents in single-use quantities and eliminate the need for any additional reagent handling or dilutions prior to use. The technology has been very broadly applicable. We can apply it to RNA, DNA, a range of small molecules. We can stabilize enzymes, antibodies, antibody conjugates, and we're exploring stabilization of more complex structures and complexes. We can build the scaffolds, as I said previously, into a range of common laboratory consumables, allowing us to integrate our stabilized reagents directly into most common analytical workflows. So I hope I've given you some sense that we believe that capillary-mediated vitrification is a powerful new paradigm for storing materials that are used in life sciences research. It's broadly applicable to a wide range of biomolecules. It's suitable for complex mixtures. We can stabilize cell lysates and also assay mastermixes. Both the stabilization process and the use of the reagents is automation friendly. We can store stabilized material ambiently and also ship them under ambient conditions. And we believe that at the end of the day, this is a technology that has a lot of opportunity to simplify operational and analytical workflows for the end users. So with that, I'm going to turn this back over to Ellen to introduce our other speakers.
Thanks, Mary. After that introduction, we're excited to have speakers from B2S Life Sciences and Assay Quant Technologies that both have tested this technology and have results to share with us. First, we'll hear from Ron Bowsher and Alyssa Cieslak from B2S Life Sciences. Take it away, Ron and Alyssa.
Good afternoon, everyone. I have some brief opening remarks. My appreciation for the importance of critical reagents has grown steadily over my many years of supporting biotherapeutic drug development in both traditional biopharma and the CRO industry. The critical reagents that we use today in quantitative assays to support PK, PD, and ADA assessments is fairly consistent, but varies project to project. These reagents include the biotherapeutic itself, antibodies, other biomolecules, and various types of antibody conjugates. Next slide. We opened our laboratory operation in 2015 to focus on the generation and characterization of custom critical reagents because of our awareness of the growing need in regulated bioanalysis. As highlighted in this slide in yellow, our core mission is the design, the generation, purification, characterization, and life cycle management of custom critical reagents. Today, the demand for high-quality critical reagents continues to grow commensurate with the increased complexity in biotherapeutics and the evolving nature of therapeutic modalities. Next slide. As shown in this slide, there's also increased awareness for the importance of high-quality critical reagents among global regulatory agencies. This fact is best exemplified by the recommendations outlined in the FDA's most recent ICH-MT guidance, ICH-M10 guidance document. Next slide. Our critical reagent business models depicted in this slide. It's a multi-step standardized process that includes design, generation, characterization, controlled inventory management, and life cycle management. Each reagent project is accompanied by a detailed reagent characterization form that's commonly referred to as a C of A. Despite immense progress in this area, we continue to face operational challenges for global distribution of critical reagents. These challenges include a requirement for frozen storage, the need for dry ice during shipments, space demands for styrofoam containers, and increased personnel requirements to support logistics function. All of these factors increase time, effort, and the cost for supporting critical reagents. To overcome these burdensome demands, we have partnered with UPCARA to evaluate their capillary-mediated vitrification technology, commonly referred to as CMV, as a cost-effective and operationally efficient alternative to traditional storage methods. CMV enables ambient temperature storage of critical reagents. In addition, it's broadly applicable and offers immense potential for improving critical reagent sustainability. At this time, I'd like to introduce my colleague, Alyssa Cieslak, who will share some of our recent CMV results for a model affinity purified polyclonal antiserum against a candidate peptide therapeutic. Alyssa?
Thank you, Ron. Hello, everyone. My name is Alyssa Cieslak. I am a scientist with B2S Life Sciences. I have been here for approximately 4 years. I work mainly with protein and biomolecule purification and bioconjugation, as well as the biophysical analysis and characterization of many reagent samples that we work with. Today, I will be discussing the data obtained during our evaluation of vitrification for storage of an antibody critical reagent at ambient temperature and the impact that may have on the binding activity, biophysical stability, and laboratory sustainability. The antibodies utilized for vitrification were generated by 8 rabbits during a 12-week immunization protocol involving injection of a proprietary peptide immunogen. Following completion of the immunization protocol, the antisera from the animals who demonstrated the most robust antibody responses were pooled and subjected to multi-step purification, including a batch purification, immunoaffinity purification, and a preparative size exclusion chromatography to remove any high molecular weight or low molecular weight impurities. A portion of the immunoaffinity purified rabbit polyclonal antibodies were shipped frozen to UPCARA for the vitrification process, while B2S retained a portion of those purified antibodies for initial characterization studies. The starting material was analyzed either as is or after mixing with the stabilization buffer at a 1-to-1 ratio. Following the vitrification process, B2S received 3 vitrified samples from UPCARA. We received a frozen sample, an unstressed vitrified sample, which was held at ambient temperature for 7 days, and a stressed vitrified sample, which was incubated at 37 degrees C for 7 days. Following the elution process, the 10-fold diluted samples all underwent identical characterization, protein quantification, purity, thermal stability, and binding activity. To assess the protein recovery post-vitrification, the concentration of the eluted vitrified samples was measured using nanodrop at absorbance A280. After accounting for the tenfold dilution, the protein yield was calculated to be greater than or equal to 97% for each of the vitrified samples relative to the frozen control. The CV across all replicate scaffolds were 2.9% for the unstressed sample and 5.8% for the stressed sample, indicating both a high yield and a consistent mass recovery. The high-performance size exclusion chromatography analysis of the purified starting material as well as vitrified samples was performed. The vitrified samples demonstrated a small shift to longer retention time as well as a broadening of the peak. However, no evidence was seen for the formation of high molecular weight oligomers or aggregates, and additionally, no fragmentation was observed. Thermal stability measurements were obtained using the Unchained Labs Uncle platform. Prior to testing, the frozen control was diluted 1 to 10 with water to match the concentration of the vitrified samples post-elution. The thermal melting and aggregation measurements were obtained during a thermal ramp from 15 to 95 degrees Celsius with a ramp rate of 0.5 degrees Celsius per minute. To determine the melting temperature of a protein, the Uncle software measures the intrinsic fluorescence of tryptophan and tyrosine residues as the protein undergoes conformational changes by way of the protein unfolding due to the thermal stress. The TM, or melting temperature value, was obtained by analyzing the first derivative of the barycentric mean of the fluorescence intensity. The melting curves for each storage condition are indicated by the serrated lines, and the TM, as determined by the Uncle analysis software, is represented by the solid vertical line on each graph. The first-order differential emission curves for each condition show a single major structural transition, and an overlay of all three conditions demonstrates comparable profiles. When compared to the frozen control, the unstressed vitrified sample and the stressed vitrified sample exhibited slightly lower melting temperatures. However, the results are within the range of values we would expect to see for an IgG. The aggregation temperatures were measured by static light scattering intensity at 266 nanometers by the Uncle system utilizing identical thermal ramp conditions. The overlaid aggregation curves for each storage condition are indicated in the serrated lines, and the TAG, or aggregation temperature onset of aggregation, is represented by a vertical line on each graph. A control experiment was performed to evaluate the impact of the stabilization buffer on the light scattering signal, and the data does demonstrate a decrease in signal believed to correspond to precipitation or large aggregates, and a corresponding decrease in the scattering intensity was observed as well. The results obtained indicate that the frozen control and vitrified samples all exhibit aggregation temperature values of 74 degrees Celsius or greater. And the vitrified materials tended to demonstrate a slightly faster aggregation rate as the temperature exceeded the onset temperature. However, neither the frozen control nor the vitrified samples show the same decrease in light scattering that is observed above 80 degrees Celsius for the antibody starting material. Next, we analyzed the binding activity. So the binding activity was evaluated via sandwich ELISA, and the binding curves obtained with the two vitrified samples overlap with the curve obtained for the frozen control. The EC50 values were comparable between the stressed vitrified, unstressed vitrified, and the frozen control, and the EC50 values ranged from 25.4 nanograms per milliliter to 31.2 nanograms per milliliter, with the EC50 values for the two vitrified arms bracketing the frozen control. The result of differing values is likely due to normal method variation. The second method for analyzing binding activity was performance of binding kinetic characterization. The binding kinetic measurements were obtained using the Sartorius Octet HTX system. And for this assay, pre-hydrated streptavidin biosensors were loaded with the biotinylated proprietary peptide at 12.5 micrograms per milliliter and dipped into the assay buffer to establish a baseline. The loaded biosensors were subsequently dipped into wells containing rabbit antibody diluted in assay buffer of various concentrations to monitor the antibody association. Following the association step, the dissociation was monitored by dipping the antibody-bound biosensors into the wells containing assay buffer. And upon analyzing the data, binding curves were fitted after subtracting reference well signals to obtain relevant on and off rates as well as the overall binding affinity. The association rate constant, dissociation rate constant, and binding affinity values were comparable amongst all three samples, confirming the antibody affinity and binding kinetics were unchanged post-vitrification, storage, and stressing. Samples stored under all three conditions demonstrate comparable binding affinity of 1 to 2 nanomolar towards the target peptide and similar on and off rates. The binding kinetics data, along with the ELISA data, indicates that the binding activity of the antibody was not adversely affected by either the stabilization process or ambient storage. The purpose of our study was the evaluation of vitrification for storage of an antibody critical reagent at ambient temperature and the impact that it could have on binding activity, biophysical stability, and laboratory sustainability. The data obtained indicates that the capillary-mediated vitrification process yielded high protein recovery with no observable impact on critical reagent binding affinity or activity towards its intended target. And while the thermal stability-based biophysical characterization data does demonstrate evidence of possible minor structural changes, additional studies would need to be performed to further understand the protein size and particle distribution pattern upon vitrification. The data indicates feasibility of CMV-stabilized unconjugated antibody reagents for use as an analytical reagent, which can be stored and shipped ambiently and are resistant to temperature excursions up to 37 degrees Celsius. This technology is a viable option for reducing costs and carbon emissions associated with cold shipping and cold storage, which overall would improve global access to critical reagent supplies. If you do have any further questions, you can reach out to Dr. Bowsher, myself, or the Senior Director of our Critical Reagents team, Dr. Sandifer Abdeen. And I am now going to pass it back over to Ellen.
Thanks, Ron and Alyssa. I think that has some really interesting implications for antibody reagents. Next, we'll hear from Gabriela Pikul from Assay Quant Technologies. Thanks, Gabriela.
Hey everyone, thank you for letting me speak today. I'm from Assay Quant. I've been there for about 2 years. I'm a scientist within the inventory management and automated workflows group. And basically, I work to implement automated workflows for kinome profiling and other assays here at Assay Quant. I'm going to be talking a little bit more about the evaluation of capillary-mediated vitrification as an alternative method for biomolecule stabilization. And I'll be speaking specifically about its application in the context of Assay Quant's phosphosense assay. So for those of you who aren't familiar with Assay Quant, we're an emerging tool company that's focused on developing a powerful technology to better understand kinase, phosphatase, and lipid kinase function. And mainly our technology is applicable to identify the most effective drugs combating cancer, chronic inflammation, neurodegeneration, and other diseases. So how do we actually do this? Well, our phosphosense platform is our main product that we offer. We develop these novel fluorogenic sensors, and they're capable of relaying information on catalytic phosphorylation and dephosphorylation via a direct measurement of these processes. And that information is actually available in real time in a continuous format. So we actually have these kinase and phosphatase assays offered as catalog products, but we also harness the technology to provide CRO testing services. So here I've included a picture of our newest edition. This is our fully enclosed automated system that we use for our kinome profiling. And why did we partner with Apkara? Well, like many labs, we have a lot of different reagents that we store to support our testing. And a lot of these reagents are temperature sensitive. We were really intrigued with Apkara's success stabilizing different types of molecules with CMV, and we were interested to see if we could apply it in the context of our own phosphosense assay. So just a brief overview of our technology and the products that we offer. We do have a wide variety of sensor peptides available. They cover up to 436 unique enzymes, and including mutants, that's up to 700 assays that we're supporting right now. You can purchase the sensor peptides either in bulk or part of an evaluation kit. Evaluation kits, technically, it's all the components that you'd need for a phosphosense assay, except kinase, at least for the moment. We also offer kinome profiling. So if you have compounds you need to determine specificity or potential off targets across the kinome, we can run your compound against 400 wild type kinases. And the cool thing is it's a continuous format. So not only can we generate inhibition data, but it's also, compound dependent kinetics that you can extrapolate or mechanism of action information that you can get from the data that we generate. As the other folks from B2S already mentioned, there's a lot of cold chain dependence throughout drug development, discovery, and for us, it's really no different, even with our CRO functionality, distributing different products worldwide. We're very much focused. A lot of our shipments go out on dry ice, especially the evaluation kits, which are pictured here on the right. And all of these dry ice shipments, they're associated with higher costs, the special styrofoam packaging that tends to be less environmentally friendly. There's a lot of increased logistical complexity here in terms of the days that you ship out domestically, internationally, and whenever you're dealing with customs, people are always concerned about delivery delays, especially with temperature sensitive reagents. Aside from that, we have a lot of reagents that we store on site. And most of the time, people aren't too concerned. It's not really on the forefront of your mind whether your freezer is going to work or not, because it usually does. But when there's power outages, suddenly it becomes a lot more high profile, the need for an alternative storage method. So in our initial discussions with Apkara, we decided on two potential applications for which we could use capillary-mediated vitrification. On one hand, we have our evaluation kits. We wanted to eliminate the dry ice shipment of these kits. And also, because this tends to be a customer's first interaction with our product, we wanted to see if we could simplify it as well, just to make it a more easy adoption, see if it works for them without the hassle of, receiving it on dry ice, storing it in a cold environment, and et cetera. And then on the other hand, protein kinases, we were interested to see if we could even actually stabilize our protein kinases and store them ambiently with the CMV technology. And internally, that would help us really simplify our storage and inventory management of all the kinases in the assays that we support, and also reduce the freezer costs and associated risks with that storage. So our typical assay, it's really made up of our phosphosense sensor peptide, our kinases, but our mastermix is the biggest thing that combines multiple reagents. We wanted to see if we could evaluate each of these different components vitrified individually and also vitrified jointly on one single scaffold. And then, barring any complications, we also wanted to see how far we could push it and whether we could also incorporate sensor peptide onto the same scaffold with our jointly vitrified reagents. From the kinase perspective, we chose just two protein kinases that we work with regularly in our selectivity and potency testing. And we wanted to evaluate those side by side compared to our traditionally stored and also our shipping control that was shipped back and forth from Apkara on dry ice. And lastly, we utilized some of the samples that were vitrified from Apkara in order to perform potency assessments against two reference compounds that we had in-house. Just very quickly, this is sort of our typical reaction setup. We have our phosphosense substrate, our master mix, and then we add either kinase dilution buffer for our blanks or kinase for our plus kinase curves. So you can see this is just a typical continuous assay format here. You can see every other row, you see blanks, and then you also see your progress curves that have kinase within them. Just highlighting our reaction conditions, you can see we do have quite a variety of different things that go into each well. And delivery of this is much simplified by having only one or two different things that you're adding to the wells rather than this whole bunch of different reagents. I will highlight that right now we use the Synergy Neo Biotech Synergy 2 plate reader. Our technology is compatible with all readers that are available with continuous reads. And then the last thing that I wanted to highlight here is really the continuous nature of this assay. You're not just getting one time point per well, you're getting multiple time points over a period. So you can really extrapolate multiple different conclusions from this. If there's time-dependent inhibition, we can often see it here already. And so moving on to some of the data that we collected as part of this pilot study, we compared different master mix storage conditions. So we had our on-site storage control. These were reagents that were stored at -80°C. These are shown in the blue. And then we also had our shipping control, which was all our reagents shipped on dry ice to Apcara and then back to AssayQuant, just to see if there were anything that occurred in transit that might be affecting the activity of these reagents. And then lastly, we had two different vitrified samples that Apcara kindly supplied for us to test. We had the individually vitrified components as well as the jointly vitrified components. And what you can see is in a continuous kinetic assay, all these different conditions perform very similarly. In fact, the reaction rates that we derived from both the shipping control and both vitrified samples overlay very, very well here. And because of the results that we received or that we tested and are shown previously, we also decided to incorporate the sensor peptide and see if we had equivalent performance with that sensor peptide vitrified alongside all our other master mix components. We see that we don't see any decrease in activity when we vitrify jointly with the sensor peptide. So we're quite confident that we're eluting the appropriate concentrations of all our components, and we are able to see equivalent results regardless of whether we have components that are vitrified or stored at -80°C in-house. So I know Mary already covered this a little bit, but it really is a very, very simple process. Just like she said, you really just rehydrate the samples. You can centrifuge them or just use them directly. And so for us, it's really a matter of minutes before we're starting to run our reactions. Moving on to the vitrification of protein kinases. So as I mentioned, we chose two samples to focus on, SRC kinase and BTK kinase, both from the tyrosine kinase family. With SRC, it's quite a stable kinase in our hands, BTK a little bit less so. So we wanted to evaluate both and see if we saw any differences with the vitrified material here. Again, we tested both on-site controls, shipping controls against vitrified samples. You'll see the on-site control here in green, along with the shipping control in yellow. And then the initial evaluation that we did was with kinase vitrified in biofix buffer, and then stored ambiently and shipped ambiently. And you can see that in orange. And so there is quite a large activity difference between the originally vitrified samples and the on-site and shipping controls. And so that was a little bit of a concern for us. We weren't quite sure if we simply weren't eluting the full amount of kinase from the scaffold, or if possibly the kinase was being denatured or otherwise dying off somehow during the vitrification process. But we spoke with Apcara and they offered a couple suggestions. And so we did another round of testing, which is the conditions are shown here in D&E in purple and blue. And basically we tried a different formulation of biofix buffer, a low binding formulation, as well as jointly vitrifying our kinases with the kinase dilution buffer that we already use in-house, but just adding those components to the same matrix that we were using for the kinase. And in both of those conditions, D and E, we do see very, very comparable results to our on-site and shipping controls. So that was very reassuring that simply it was a matter of kind of customizing the buffer. And we do now have several options to proceed with. Hopefully, at some point, we'll be able to expand the use of this to our full panel of kinase assays. And lastly, we did do some potency testing with 2 reference compounds against BTK kinase, ibrutinib and sunitinib. We were able to show that IC50s are within 2-fold for these 2 control compounds, regardless of vitrification method, as long as we were able to get the same level of activity with the kinase. And just to wrap up some key takeaways. So after evaluating Apkara's CMV technology, we do have a path forward to ambient phosphosense evaluation kits and reagent packs. That was pretty much the biggest gap that we saw that Apkara could fill for us. And I'm happy to say that there is definitely a path forward there. We also have potential for reduced shipping cost and minimized environmental impacts with the elimination of dry ice shipments. And then we already have a powerful technology, but when we partner with Apkara, it can be even easier and quicker for us to bring that technology to our customers and also to use it internally. We do have many opportunities now that we can see for how we might streamline on-site storage of reagents and consequently overcome all the time, space, and cost constraints that are associated. And lastly, this one is sort of my personal favorite since I have a background in automation. Apkara shared that they have the new scaffold, different new scaffold modalities that are available. So they have essentially, you can embed a scaffold in 96 different wells on a filter block. For us, that would be great because it's actually automation compatible. And a lot of our higher throughput processes are actually moving in that direction. And so we could see a lot of potential with this, especially in our higher throughput profiling processes. So with that, I wanted to thank everyone who helped contribute to this effort, especially Khan from AssayQuant. She generated a lot of the data that I shared here today. And also Sherry from Apkara, who went back and forth with us in designing some of the experiments and also provided all of the samples that we tested. And if you want to contact us for more information or are interested in how we might help you, that is our email, hello@assayquant.com. And if you have any further questions specifically regarding these initial evaluations with Apkara, feel free to reach out to me as well. And with that, I'll pass it back to Ellen. Thank you.
Thank you, Gabby. And thanks to all our speakers for your presentations. We do have some time for some question and answers. So please remember to post your questions in the Q&A tab that sits at the bottom of your Zoom browser. I'll go ahead and begin. Mary, this first question is a follow-up question for you about some specifics about molecules. 2-parter. Can you maintain the native structure of proteins using CMV? And how stable is RNA with CMV at room temperature?
So starting with the question about proteins, I hope that we've shown you that we're doing a lot of testing to make sure we can maintain the activity of proteins. We talked about maintaining the binding activity of antibodies and the enzymatic activity of kinases. That data indicates that we are maintaining the native structure of the proteins. If we were not, we would be losing their function. We also, in the thermal melting data that Alyssa shared, we're not seeing any evidence of denaturation and we're not seeing any evidence of aggregation. We do see some minor shifts in the hydrodynamic radius, which we actually think are a result of an interaction between the protein and the stabilizers. So we think that there's we know our stabilizers are helping to replace hydrogen bonds, and that could be helping to or could be impacting the hydrodynamic radius. So we think that's what we're seeing. We're going to do some more studies to evaluate that, but we're clearly maintaining function, and that's really what we're most interested in doing. And then the second question was about RNA. We have successfully stabilized purified RNA at 37 degrees and higher temperatures. I believe we've stored RNA for more than 3 months at 37C with no loss of transfection efficiency. And so we're getting great results with mRNA. We've also stabilized mammalian cells at 55C for several days and shown that we can extract the RNA and do deep sequencing, and we get comparable data to freshly cultured cells.
Great, thank you. My next question is a follow-up for Alyssa. This is with regards to the elution peaks that you showed us in the HPSEC profiles. Why is a broadening of the peak or differences in retention time observed in those profiles?
So our hypothesis for the changes in the size exclusion profiles is really a result of the possible interactions of the protein with the sugar molecules in the buffer during the vitrification process, kind of like Mary had said earlier. The data does clearly show that there is no aggregation or fragmentation, so there might be a slight structural change causing changes within the polydispersity of the sample itself.
Let me just add on to that, if I may.
Yeah,
the
absolutely.
other thing that
Of
we
course.
were seeing, which Alyssa pointed out, is that when we do the thermal aggregation studies, even once you've rehydrated the protein, they appear to be more resistant to aggregation and precipitation. So that's a very interesting finding that we want to follow up, because it suggests we may have some benefits in terms of the post-rehydration solution stability.
Right. My next question is for Gabby and Mary. We'd love to hear more, I think, about the adaptability to various master mixes. So I suppose first, what other types of master mixes has this been applied to? And Gabby, if you could speak to sort of the individual compound stability after vitrification. There was one question about DDT in particular.
Gabby, do you want to start, or would you like me to start?
You can go ahead and start and I'll follow up.
Great. So that's a great question. We're very interested in stabilizing master mixes. We believe this is a technology that can help simplify analytical workflows. In addition to the master mix that we prepared for Gabby and her team, we have prepared PCR master mix using both traditional Taq polymerase and hot start polymerase. We have produced some other master mixes of, I would call them more multiplexed reagent master mixes for the Luminex platform, both looking at conjugated beads and in the antibody cocktails that are used for detection. And we've looked at some master mixes that are used to do mix and read ELISAs by another customer, where they had some antibody conjugates and a small molecule substrate that they wanted to co-formulate, but were having challenges finding a single formulation where they could stabilize all of the components using lyophilization. The process is pretty amenable to additives. We can add EDTA, DTT, salts, pretty much any buffer component that would be needed. And the process is tolerant of some quantity of glycerol. We can't tolerate infinite amounts of glycerol, but we can accept some quantity, and we can also incorporate detergents if needed.
Yeah, Gabby, what was your experience with sort of working around the various compounds in your master mix?
Yeah, so we did, I highlighted a lot of the functional data with our assay in my presentation, but we also used UV spectroscopy to quantify some of the eluted concentrations of reagents. And we found that to be quite compelling. The data agreed within experimental variants that we would expect. And so we mostly focused on ATP because we see that impacting our assay and our rates a lot more. But we did find with successive elutions, we saw less than 1% of the target concentration. Again, that's not so much speaking to the stability, but I think in future follow-ups, we would probably look more at the stability of both DTT and the rest of the reagents that we use.
Great, thank you. One thing I was really interested by at the end of your presentation, Gabby, was your mention of automation and scaffolds. I would love to hear from the users of this technology, what type of scaffolds you worked with, and then Mary, perhaps what we can sort of expect of scaffold sizes and types in the future for folks to use.
So I can just quickly speak. So for our initial evaluation, we actually just used 15 and 50 mil conical tubes. Those would be sort of the pack sizes that we would anticipate using with our reagent packs. But again, the team at Apkara did share that they have the 96 well blocks. And so, yes, I'm very excited about the potential with those.
Yeah, so we're actually developing a number of scaffold materials. And there are different reasons for using different materials. But if you think about a scaffold of being similar to a sponge, if you want to hold a small volume, you would like to have a thinner sponge. So there's not any excess holdup volume. We don't want to lose material or have material get trapped in that sponge. If we need to stabilize a lot of material, we'd like to have a thicker sponge. And then the other factor we think about is the surface area of the scaffold. The larger the surface area, the higher the mass of the biological reagent that we can store on a single scaffold. So we typically work with customers and understand from them what is the aliquot size that they're interested in, what's the mass of protein or RNA or DNA, and what's the volume of solution that they want post-rehydration, and we'll use that to select the appropriate scaffold material. We can then build that scaffold into an integrated device. That could be a 50 mil tube. As Gabby mentioned, it could be a microcentrifuge tube. It could be a 96 well plate. It just depends on the workflow preference.
Interesting. Yeah, I'm looking forward to learning more about the scalability of this technology. Sounds like there's some exciting developments in the works. But I am going to go back to the question previous to that, actually. Mary, you mentioned the conjugated biomolecules. We have a question from the audience asking if this application has been used on fluorophore conjugated antibodies, particularly for flow cytometry. If so, they're curious about the stability of binding and how that's maintained due to changes in temperature incubation as the nature of the assay.
That's a great question. We've done a lot of work with conjugated antibodies in both an ELISA format and then also more recently in flow applications. We've looked at enzyme conjugates, fluorophore conjugates, and then more recently, we're starting to look at some tandem dyes. And we are seeing very comparable performance between the vitrified reagents and frozen reagents. So we actually think this could be a great solution for simplifying flow workflows and enabling researchers to make stable custom panels that they don't have to prepare every day that they want to use them.
Sorry to keep bothering you about specific types of molecules, but we have another question here. Has anyone used this approach for stabilizing vectored vaccines or viruses?
That's a great question. Anything about vaccines I love to talk about. We've done a little bit of work looking at a lentivirus. We haven't optimized at this point, but that is definitely something that's on our radar and something we're really interested in getting some feedback about what would be an interesting virus to work on. And then we'd love to find some partners who have all the right analytical tools to really fully characterize a stabilized vaccine.
Great. I think I have time for one more question, but I'd kind of like to pose it to the whole panel if you have anything to add. All four of our speakers, Mary, Alyssa, Gabby, and Ron, spoke to sort of the economic and sustainability impact of using a room temperature stabilization technology for reagent storage and shipping. One question in particular has been about cost analysis. If this technology has been compared to other stabilization techniques, I'm going to guess most likely interested in lyophilization in particular. But if anyone wants to speak to what they think the impact might be, and how to sort of analyze the cost impact as well. Mary, why don't you go ahead and get started?
Sure, I'll happy to start. So there's actually two parts to your question. So one is, what's, I think, the cost of the stabilization process. And I'd love to say it's, X cents per container, but the process is really customizable. So the cost really depends on how the customer wants to integrate the scaffold. Do you want a very simple unstructured scaffold? Do you want something that's built into a 96 well plate? That has a big influence on the cost. There are many opportunities for cost savings comparing this technology to lyophilization in particular. One of the biggest is simply time and labor costs. Our equipment is less expensive, and our cycle times are on the order of an hour. And so when you compare that to a lyophilization process, which can take anywhere from 12 hours to 72 hours, and you think about the throughput capability, there's huge possibilities right there for cost reductions. Additionally, if we can reduce the amount of cold storage, cold shipping, there's a lot of cost savings just around, minimizing the number of ultra cold CTUs that exist in the laboratory. So it's really something we have to work with the customer on, but we do believe this is going to ultimately be very cost effective when you look at it holistically. Ron, I don't know if you've got any comments about it, if you agree with that assessment.
Yeah, I agree fully with the potential for cost savings. At B2S, we think about our clients and being able to give them access to critical reagents in a small box shipped at ambient temperature. Those are the kinds of cost savings that you can pass on to the customer and to help control, just the cost of doing business. So we're very excited about the potential of using this kind of broadly for a lot of our antibody and antibody conjugates. Yeah.
I suppose on the user end, Alyssa and Gabby, if you could speak to the reconstitution process and actually using the lyophilized material, what that impact looks like in the lab.
I would say for me personally, it would cut down on time spent in the lab trying to thaw materials, because the elution process is very simple with the scaffold, and having to delay a start time waiting for a material to thaw on ice or at room temperature. I think that it would cut down on the saving time as far as a consumer and the time that I have to spend in the lab waiting rather than actually performing the experimentation.
True. Gabby?
So a lot of the same sentiments that Alyssa expressed. It is very, very easy to use. For us also, we spend a lot of time aliquoting a lot of our kinases, and sometimes that can be tedious. There's a lot of, just consumable equipment involved there too. So potentially this could be another way to minimize that as well.
I think those were all fair points. That is all the time for questions we have today, but if we have additional questions, we'll follow up with you all that have submitted them to the Q&A. So we'll certainly respond and get back to you. If you continue to have additional questions about Apkara's technology, please reach out online at apkara.com or through email to info@apkara.com. Also keep an eye on your inbox for announcement of upcoming Apkara webinars. I want to thank again our speakers, and we thank you all for attending today's webinar. Goodbye and take care.